So when it comes to gene therapy, fixing a broken gene is really only half the story. The real question is, how do you actually deliver that fix to the right place? Today, we're going to look at the tiny, microscopic mail carriers that make it all happen.
Okay, imagine your DNA, your body's instruction manual, has a tiny typo. Just one little spelling mistake that's causing a huge problem, a serious disease. Gene therapy is all about correcting that typo. But you can't just inject the right spelling and hope for the best. You need a delivery system.
And this is where things get really, really cool. Scientists have basically hacked viruses. They've taken these things that normally make us sick and turned them into microscopic mail carriers. Their whole job now is to carry a precious package, the healthy gene, and deliver it right where it needs to go.
Now, in this world of genetic mail, there are two real heavy hitters that scientists use all the time. AAV, which stands for adeno-associated virus, and lentivirus. The biggest difference between them isn't just their names, it's how they handle that genetic package once they've dropped it off.
All right, let's get into it, starting with our first carrier, AAV. The best way to think about AAV is as a specialist. It's designed for cells that have kind of settled down for good, the ones that aren't really dividing anymore.
So AAV is the go-to choice for diseases in places like the brain, the eyes, and our muscles. And why is that? Well, it's because those parts of our body are full of cells that, once they're mature, they just stop dividing. They're permanent residents, and that's the exact kind of cell AAV is perfect for. And what's really amazing is how AAV makes its delivery. When it gets inside the cell's command center, the nucleus, it drops off the healthy gene. But here's the key. That new DNA doesn't get woven into the cell's main chromosomes. Nope, it just hangs out nearby as a separate, little floating loop called an episome. For a cell that isn't dividing, this is awesome. That little episome can just sit there for years doing its job. This strategy works perfectly until it doesn't. You see, if you try to use AAV in a cell that is still growing and dividing, you hit a pretty big wall. It's something called the dilution problem. Think about it. When a cell divides, it makes a perfect copy of all its chromosomes for the new cell. But that little floating episome, it's not part of the main blueprint, so it doesn't get copied. It basically gets left behind. So with every division, the therapy gets weaker and weaker until it's just gone.
And that brings us to our second mail carrier, lentivirus. If AAV is a temporary guest, lentivirus is moving in for good. It's built for cells that are constantly on the move, always growing and dividing. This is the absolute workhorse for therapies that target dividing cells, like the stem cells in our bone marrow that make all our blood, or our immune cells. In fact, lentivirus is the star of the show in those incredible CAR-T cancer therapies you may have heard about.
See, a lentivirus doesn't just drop off its package. It has a key to the cell's most secure vault, the chromosome itself. It finds a good spot, opens up the DNA, and literally stitches its genetic cargo right into the host cell's own blueprint. And that right there, that's the superpower of lentivirus. Because once that healthy gene is part of the blueprint, it's there forever. Every single time that cell divides, the therapeutic gene gets copied right along with it. It's a permanent fix that passes down from one cell generation to the next.
Okay, so we have two totally different approaches, right? The temporary episome versus the permanent integration. Now that we've met both of our carriers, let's put them head to head and really break down the differences.
So the main thing is clear, right? AAV is for non-dividing cells, lentivirus is for dividing cells. But look, there are a couple of other really important differences here, how much they can carry and how they stack up on safety. Let's talk about cargo space. Some genes are pretty small, but others are huge. AAV is kind of like a compact car. It's super efficient, but it can only carry a small package. Lentivirus, on the other hand, that's your delivery truck. It's got a much larger payload, so it can carry those big, complex genes that AAV just can't fit.
And finally, safety. This is a big one. Because AAV's little package stays separate from our DNA, it's considered to have a very clean safety profile. Lentivirus, because it's literally cutting and pasting into our genome, carries this small theoretical risk of something called insertional mutagenesis. It's basically the tiny, tiny chance that it could accidentally insert the new gene in the wrong spot and mess up an important healthy gene.
So, after all that, you might be thinking, okay, which one's better? But that's really the wrong question. It's not about which one is better overall. It's about picking the perfect tool for a very specific job.
It all boils down to one simple golden rule. The cell type determines the carrier. If your target is a cell that's going to be there for life, like a brain cell, use AAV. But if you're targeting a cell that needs to multiply and grow, like a blood stem cell, you absolutely need the permanent fix that lentivirus provides.
I mean, these technologies are incredible. They're completely changing the game for diseases that we used to think were untreatable. And really, this is just the beginning. So, as we wrap up, here's something to think about. What major problem do you think these microscopic mail carriers should be sent to solve next?